Hello everyone, so I'm back with my next video. In this video I'll be talking about immunity and the different types of immunity. Now what is basically immunity? We know that we are surrounded by germs. The air we breathe in, the water we drink, the different things that we touch, none of these are free from germs. The entire world of invisible microbes is there. However, though we come in contact with so many germs on a daily basis, we hardly fall sick. Reason is there is this immune system which is continuously protecting us. So when we talk about immunity basically can be classified as innate immunity and acquired immunity. When you are talking about innate immunity, these are inborn immunity. You don't have to acquire them. So from birth, by the virtue of birth, you get these kind of immunity. So what are these immunity? They can be physical barriers, physiological barrier, cellular barrier, and cytokine barrier. Now when we talk about physical barrier, it's given there in your book. So when we are talking about physical barriers, it includes our skin. Skin, you know, the first barrier which the germs faces when they try to enter our body. Then we have the mucus coating in our nose, then in the GIT, gastrointestinal tract, urinogenital tract, that is the urethra. So all these helps in preventing the microbes from entering. Next we have physiological barrier. Physiological barrier means any kind of chemical which is secreted as a result of body process. So like the hydrochloric acid in your stomach, the saliva, you have heard about lysozyme enzyme present in saliva, tears from eye, all these are physiological barrier. You must have seen that when any animal gets hurt, it licks the wound because the saliva contains antibacterial properties. Next we have cellular barriers. Cellular barriers are some kind of special cells which actually help in directly fighting with the germs. Like you have PMNL, polymorphonuclear leukocytes, neutrophils. Neutrophils are kind of WBCs. They have different morphological shaped nucleus. So they are called polymorphonuclear leukocytes. Similarly, the other cells which provides us with cellular immunity are like the macrophages. Now where do you find macrophages? If you recall, you have studied about areolar tissue in class 9. Areolar tissues are connective tissue. Suppose there is your kidney and there is your body wall. So the kidney will be held in its position by these fibrous tissue, and in this fibrous tissue there are some amoeba shaped cells which are called macrophages. So all these cells, they will directly engulf the germ, thereby providing the immunity to your body. And then we have cytokine barrier. In cytokine barrier, what we have, like if any of your cell is infected by a virus, then that cell will produce some proteins, and those proteins are called interferons. Those interferons will be protecting your neighboring cells from infection. So that is called cytokine barrier. So these are non-specific kind of barrier. They don't provide protection to your body against specific germ. However, there is another kind of immunity which is called acquired immunity. Now what is acquired immunity? Which you acquired during your lifetime on exposure to germ. So these acquired immunity is of two types: AMI, antibody mediated immunity, and cell mediated immunity. Antibody mediated immunity is also called humoral immune response because the antibody molecules, protein molecules, are found in blood plasma. That's why it is called humoral response. These are provided by B cells and this is provided by T cells. Now what is B cells and T cells? To understand this, let us do a quick recap of the composition of blood. So blood, you know, it consists of plasma, 55% blood volume, blood corpuscles, 45% blood volume. The blood corpuscles are of three types: red blood cells or erythrocytes, platelets or thrombocytes, and white blood cells, which are leukocytes, can be further classified as agranulocytes and granulocytes. This is based on whether granules are present in the cytoplasm or not. So in our granulocytes, the cytoplasm is clear, no granules are there. There are of two types, lymphocytes and monocytes. Granulocytes, they have fine granules in the cytoplasm, and the nucleus is multilobed. They are of three types, neutrophil, basophil and eosinophils. Now, neutrophils are the most abundant cells. WBC is 40 to 60% and lymphocytes are 20 to 40%. So, these lymphocytes are the main protective cells. Why? Because these lymphocytes are further classified as B lymphocytes and T lymphocytes. So B lymphocytes are called B cells, T lymphocytes are called T cells. So B lymphocytes, they are of two types, plasma B cells and memory B cells. And T lymphocytes are of four types, cytotoxic T cells, suppressor T cell, helper T cell and memory B cells. Memory T cell. Now why they are called B cells and T cells? Because these cells will mature in the bone marrow. These cells will mature in the thymus. Thymus is a lobular organ present just above the heart. It is very big and highly active during birth. Upon reaching adolescence, it shrinks and becomes more or less very much not so indistinct, becomes indistinct, you can say. Okay. So now how these cells will provide us immunity. So I told you that the kind of immunity, AMI, it is provided by B cells or antibody mediated immunity. Now why it is so? Because you see, the plasma B cells on getting activated, okay, these will mature in bone marrow, these will mature in thymus, but only maturation will not do. To get activated, they get activated in lymph nodes, that we'll discuss in the lymphatic system. Okay, so plasma B cells, they will end up producing antibodies. Antibodies are protein molecules. A structure of antibody molecule is given in your textbook. So we can have a look at the diagram of this antibody molecule. It is a protein molecule, so it has polypeptide chain, 4 polypeptide chains. The inner chains are heavy chain, outer chains are light chains, and the top portion where you have found antigen binding site, this is the variable region and this is the constant region. Variable region because they can change the configuration to bind with the germ, and the germ is called antigen. So these are the light chains and these are the heavy chains. Both are connected by disulfide bridges. Disulfide bridges you can see. And another thing which you can see, these ends are written as C and these end is written as N. Why it is so? Because there is a protein chain. Proteins are made up of amino acids. Amino acids, you know, one end will be carboxylic and another end will be amino group. So this side is the amino group, this side is the carboxylic group. So these are typical antibody molecule. So antibodies are classified into 4 groups: IgM, IgD, IgG, IgA and IgE. So IgM, these are very big size antibody molecules, and their binding site is so being big in size, they can easily bind to germ, they can easily identify germ. So they provide the first primary response on when your body gets infected by any germ. IgD, the function is still not so well defined. However, scientists say that they activate the B cells. IgG, the most abundant antibody molecules, 70 to 75% are IgG. Some of the IgG also pass from the placenta to the fetus, providing protection. IgA is abundantly found in colostrum. These are secretory antibody. They are found in your mucus, in saliva, in tears, and they provide protection to the newborn child. And IgE, it causes a kind of allergic reaction and also help in killing worms. So all these are higher level things. However, Ig means immunoglobulin. It's a protein, immunoglobulin, and the different peptide chain, their structure, depending on that, we have these 5 groups. So that is how we get the antibody mediated immunity. Plasma cells gets activated, and upon getting activated, they will produce the antibodies which will combine with the antigen and neutralize the antigen. Now how these antibodies are formed? Okay, before discussing this, we'll discuss about cell mediated immunity. Cell mediated immunity, see, we have four types: cytotoxic T cells. Cytotoxic T cells, how they provide protection to your body? They do frontal combat. They directly fight with the germ. They will create pores in the germs so that water can enter the germ. The germs will swell and burst open. That is how the cytotoxic T cells, they kill the germ. Suppressor T cell, now you see, your immune system is fighting the germs, which are cells. Your body also has cells, but your immune system is not destroying your cells. Why? Because your immune system is able to recognize between self and non-self. That, so keeping your immune system under check, under control, that is done by suppressor T cell. If these cells gets damaged, your body will suffer from autoimmune diseases. You have helper T cells. So helper T cells will get its role more in HIV/AIDS discussion. Helper T cells actually help plasma B cells to mature, and only then plasma B cells will start producing antibodies. So they help, that's why they are called helper T cells. And memory cells, whether it is B cells or T cells, they help in retaining the memory of the germ, the information of the germ. So since here the protection is provided directly by the cell, that's why it is called cell mediated immunity. It plays a very big role in organ transplant. You know that you cannot simply donate organ or take organ from anyone, because suppose you take organ, kidney or liver from some another person, then your body will regard that particular organ as a foreign body, and these cells, that CMI, cell mediated immunity, will reject or will kill those cells, and the organ will be rejected, it will get destroyed. That is called graft rejection. So many compatibility factors are checked before organ donation is done. Now how these antibodies are formed? Based on that, so that's your AMI and CMI. So I hope it is relatively clear. So how antibodies are formed? Based on that, we have two types of immunity. We have active immunity and passive immunity. Active immunity is when your body will be producing the antibodies on exposure to the germ or antigen. For example, you know, when you develop chicken pox, second time you do not develop chicken pox. Why it happens? Because memory cells are formed, your body has been exposed to the germ and memory cells are there. So next time when there will be more, again the infection will try to enter, those memory cells will rapidly protect your body. So that is called active immunity. And this active immunity, it is used in the process of vaccination. The principle of vaccination I will be discussing. So that is related with active immunity. Passive immunity means when your body does not have time for getting exposed to germ and producing the antibodies, like a newborn child. Newborn child is relatively vulnerable to the germs around him or her. So what happens? The mother's colostrum, it contains lots of ready-made antibodies, IgA. So feeding the baby with mother's milk provides ready-made antibodies. So that is called passive immunity. Similarly, like snake bite, you cannot wait for your body to produce antibodies getting exposed to it. So it needs required immediate treatment. So preformed antibodies are injected. So similarly, tetanus toxoid that we take. So all these are examples of passive immunity. Preformed antibodies are directly injected. So when we come to vaccination and immunization, so you know you have vaccines available for different diseases like hepatitis B. Now huge research is going on throughout the world, rapid research for finding corona vaccine. Then your polio vaccine, smallpox has been totally eliminated. So vaccination, let's give you a brief idea about the history of vaccination. Vaccination was discovered by Edward Jenner. So Edward Jenner, during his time, smallpox was a deadly disease, and a relatively less deadly disease was cowpox. He observed that people who were suffering from cowpox, they did not suffer from smallpox. So what he did, he took the pus, because when pox is formed, you know, some blisters are formed with pus inside. So he collected pus of cowpox and he injected into a small child, and the child developed cowpox and very soon recovered from it. So after a few weeks, he collected pus of smallpox, which was much more deadlier, and he injected into the same child, but the child did not develop smallpox. So that was how vaccination developed. Nowadays we have fourth generation, fifth generation vaccines. These are highly refined molecules. It's not only germs. So what vaccine basically are? Vaccines are germs of or the pathogen of that particular disease in a much weaker or killed, or any product of that particular pathogen which is injected into your body in safe doses, so that your body does not develop the disease, but it can stimulate the production of memory cells, so that next time, so that's the primary response. So next time when more active form of the germ will infect your body, so there can be a much more vigorous secondary response and your body will remain protected. So the vaccination is a case of active immunity. Your body is being exposed to germ and your body is producing the antibody and being prepared for future consequences. And this overall process of developing the immunity of your body against different diseases by taking different vaccines is called immunization. So this is all about the initial concept of immunity, AMI, CMI, then the different types of lymphocytes, and active and passive immunity. So in the next video, I'll be discussing about the allergic responses of the body and the immune system in our body, and also something about autoimmunity. Thank you.